Okay, let's unpack our first medical mystery for you today. Shock and DIC by Gram-OB bacteria.
Right, and what's truly fascinating here is the precise molecular mechanism, the trigger. Lipid A of LPS macrophage activation, TLR4-CD14, complement activation, tissue factor activation. It's essentially a specific part of the bacterial wall acting like a massive alarm bell for the immune system.
Yeah, and that alarm can spiral into something really terrifying. I remember my grandfather, he was a pretty sturdy guy, just had a routine prostate procedure. A few days later, though, he started feeling unusually weak. Like really weak. And within hours, he was shivering uncontrollably, his skin felt clammy, and he was confused, sort of muttering nonsense. By the time we got him to the ER, his blood pressure was dangerously low. I mean, classic septic shock, right? The infection was just overwhelming his organs.
Absolutely. That's the systemic response.
And the doctors explained it. They said the lipid A from those gram-negative bacteria was like this hyper-alert button for his immune cells, the macrophages, specifically through pathways like TLR4 and CD14. This kicked off a huge release of inflammatory chemicals that just plummeted his blood pressure, that hypotension.
Right, and that same massive immune activation, it also directly hits the clotting system. That explains the DIC part.
Exactly. They kept mentioning DIC, disseminated intravascular coagulation. His blood wasn't clotting right. I actually saw tiny red spots, petechiae they called them, all over his arms.
Yeah.
And blood just kept oozing from where they drew his blood.
Yeah, that's characteristic.
It was terrifying. Pale, cold, bleeding from one place, but supposedly clotting everywhere else internally. It's because that lipid A had also activated tissue factor, setting off widespread microclotting that then, consumed all his clotting factors.
Leading to that paradoxical bleeding you mentioned, consumptive coagulopathy.
Yeah. This whole cascading crisis, the organ problems, kidney, liver stuff, confusion, all from this one bacterial component. They rushed him to the ICU, fluids, strong empiric antibiotics, meds to keep his pressure up. Finding and treating that underlying bladder infection was key. Okay, moving on. Next up, we're diving into prosthetic device infection by S. epidermidis.
Ah, yes. The core mechanism here, and what makes these so tricky, is biofilm production.
Biofilm. Okay, so what does that actually mean for someone, say, with a new joint? My grandmother got a new hip, right? Everything seemed fine for the first few weeks, but then she started getting this nagging low-grade fever, just wouldn't go away.
Common presentation, subtle.
Yeah, and tired all the time. Her hip just ached more than it should. But interestingly, not a lot of redness or swelling, so it wasn't obvious. The doctors were a bit puzzled at first. Eventually, they took samples from around the hip joint and found Staphylococcus epidermidis.
A common skin bug, usually harmless, but notorious on foreign materials.
Right. Her surgeon explained it just like that. Said it's a skin bug, loves to stick to artificial parts like her hip implant, and it produces this sticky protective layer. Like a microbial fortress, he called it. A biofilm.
And that biofilm is absolutely key. It's a physical shield. It makes the bacteria incredibly resistant to the body's immune cells and, crucially, to many antibiotics. That's why these infections can be so insidious, so chronic. Just low-grade fever, malaise, maybe some localized pain, sometimes for months.
Exactly. He really emphasized that this slime layer, the biofilm, made it really hard for antibiotics to get in and actually work. So she ended up needing another surgery to replace the infected hip joint entirely. Plus months of strong IV antibiotics, vancomycin, I think.
Often vancomycin, yes, because S. epidermidis is frequently methicillin-resistant. Device removal is often necessary.
It was just a long, painful road, all because of this tiny germ that could build its own fortress. Really shows the challenge when bacteria get entrenched like that. Okay, next. Endocarditis, secondary to S. sanguinis.
Right. If we connect this to the bigger picture, the specific mechanism here involves Dextrans, biofilm production, that bind to fibrin-platelet aggregates on damaged heart valves.
Dextrans. Okay, let's unpack that. My uncle, he always put off going to the dentist. Hated it. Plus, he had rheumatic fever as a kid, which left him with a dodgy mitral valve. So a classic damaged valve situation.
Predisposing factor right there.
Totally.
Right.
So years of avoiding cleanings. He finally goes for a root canal. A few weeks later, he starts feeling really tired, just wiped out. Then gets a low-grade fever that just lingers. His wife, she's a nurse, noticed these tiny, painful red bumps on his fingertips.
Osler nodes. Classic sign.
Yes, she recognized them right away. Osler nodes. Told him to get checked out. He goes to the doctor, they listen, and boom, new heart murmur.
Through murmur, fever, Osler nodes.
Yeah.
That's highly suggestive of subacute endocarditis. You'd confirm with blood cultures, maybe an echo.
Right, and the blood cultures came back positive for Streptococcus sanguinis, which the doctor explained is a totally normal bacteria found in the mouth. But because of the dental work, maybe just his poor hygiene before that, it got into his bloodstream, bacteremia. And it found his old, damaged mitral valve. The sanguinis makes these sticky things called dextrans.
Which act like glue.
Exactly. They let the bacteria stick to these little fibrin-platelet clumps that can form on damaged valves. It's like it sets up camp.
Forms of vegetation, essentially, a biofilm on the valve.
Leading to the inflammation, the infection,
Yeah.
endocarditis. He needed a long course of IV antibiotics, ceftriaxone I think they used. There's a huge wake-up call about dental hygiene, especially for him with his valve history. Prophylactic antibiotics before dental work are key for high-risk folks.
Absolutely. Shows how something seemingly minor, like oral bacteria, can cause major problems in the right or wrong circumstances.
Okay, our next deep dive is pseudomembranous colitis, secondary to C. difficile.
C. diff. The mechanism here is very direct, very impactful in the gut. Toxins A and B damage enterocytes, watery diarrhea.
Yeah, and it can be brutal. My aunt had a minor surgery, nothing major, but they gave her antibiotics afterward, preventatively. A few days after coming home, she developed just awful, frequent watery diarrhea.
Yeah.
And severe abdominal cramps. Plus a fever, felt totally exhausted.
Classic C. diff presentation, especially after antibiotic use.
Her family doctor suspected it straight away because of the antibiotics. They did the stool test, looking for the toxins.
Toxins A and B, yep. PCR or antigen tests are standard.
And it came back positive. The doctor explained how the antibiotics she took had basically wiped out a lot of the good protective bacteria in her gut, which allowed this other bacterium, Clostridioides difficile, it's a spore former, to just take over, multiply like crazy.
And once it proliferates, it releases those toxins. Toxin A is more of an enterotoxin, causing fluid secretion, hence the watery diarrhea. Toxin B is a potent cytotoxin, directly damaging the intestinal lining cells. That damage leads to inflammation, cell death, and sometimes those characteristic yellowish plaques, the pseudomembranes, on the colon surface.
That explains the severe pain and everything. She ended up on a different antibiotic, oral vancomycin, specifically to target the C. diff.
Right. Vancomycin or fidaxomicin are first-line treatments now. Discontinuing the original offending antibiotic is also important if possible.
It took weeks, honestly, for her gut to get back to normal. She said the recovery was almost worse than the surgery itself.
It can be really debilitating. And recurrence is a problem, too.
Definitely shows how disrupting that delicate gut microbiome balance can let opportunistic bugs cause serious trouble. All right, moving on. Let's talk about diphtheria.
A historically feared disease, thankfully rare now due to vaccination. The mechanism of its potency is quite elegant in a destructive way. Exotoxin inhibits protein synthesis via ADP-ribosylation of EF2.
Elegant but destructive is right. When I was a kid, my great aunt told us this terrifying story about her younger brother. This was way before vaccines were common. She said his throat started aching. Then this awful grayish-white film, like a membrane, spread across his tonsils. It stuck fast, apparently hard to remove.
The classic pseudomembrane.
Right, and he could barely swallow or breathe. His neck swelled up massively. She called it a bull neck.
Due to the severe lymph node swelling, the cervical adenopathy. That triad pharyngitis, pseudomembrane, bull neck is characteristic.
The doctor back then apparently said it was the bacteria's poison just shutting down his body's ability to heal.
And that poison is the diphtheria exotoxin. It works by chemically modifying and inactivating a crucial protein called elongation factor 2, EF2. EF2 is essential for building proteins in our cells. So the toxin effectively halts protein synthesis, leading to cell death.
Which explains the local tissue damage, like the pseudomembrane, but also the systemic effect.
Exactly. The toxin gets absorbed and can damage distant organs.
Yeah, he developed heart problems soon after myocarditis as a well-known, often fatal complication. And nerve issues, too, polyneuropathy. It was just a brutal illness, left him weak for ages. She always hammered home how lucky we were to have the DTaP, Tdap vaccines.
Which work by generating antibodies against the toxin itself. It's a toxoid vaccine. Treatment also involves antitoxin and antibiotics like erythromycin or penicillin.
A stark reminder of vaccine power against toxin-mediated diseases.
Yeah.
Okay, next up, the virulence of M. tuberculosis.
TB. It's incredibly successful as a pathogen, partly due to its complex virulence factors. A key dual mechanism is cord factor activates macrophages, promoting granuloma formation, induces release of TNF sulfatides, surface glycolipids, inhibit phagolysosomal fusion.
Okay, so two main tricks up its sleeve. Let's unpack how that plays out. I actually had TD myself years ago after traveling somewhere it was common. For months, I just felt One down, this nagging cough that wouldn't quit, drenching night sweats, lost weight without trying.
Classic symptoms of active pulmonary TB.
My doctor diagnosed active TB, Mycobacterium tuberculosis, and they explain that this bug is so tricky. Part of it is this cord factor.
Which is a glycolipid found on the surface of virulent strains. It causes the bacteria to grow in these characteristic serpentine cords, visible under a microscope. Functionally, it's toxic, and it strongly activates macrophages, driving the formation of those granulomas, the walled-off inflammatory lesions in the lungs. It also triggers TNF-alpha release, contributing to fever and weight loss.
So it helps build the granuloma fortress, but then there are the sulfatides.
Right. These are other glycolipids on the bacterial surface. Their main job seems to be preventing the phagosome, the little pouch the macrophage uses to engulf the bacterium, from fusing with the lysosome. The lysosome contains all the digestive enzymes that should kill the bacterium. By blocking this fusion, the TB bacteria can essentially survive and even multiply inside the macrophage, hidden from the wider immune system.
Wow. So it builds a fortress, and then it disarms the guards inside the fortress.
Pretty much. That ability to survive within macrophages is key to its chronic nature and latency.
It totally made sense why treatment was such a long haul. That multi-drug regimen, RIPE, rifampin, isoniazid, pyrazinamide, ethambutol, for months.
Absolutely necessary to overcome these defenses and prevent resistance.
A fascinating example of complex bacterial strategies for survival and causing disease. Our next topic is tuberculoid leprosy.
Leprosy, or Hansen's disease. Here, the host immune response is really the star of the show, determining the clinical picture. The mechanism for the tuberculoid form is Th1 immune response, mild symptoms.
Th1. Okay. So what does that mean in practice? My neighbor, an older gentleman who grew up in a rural area overseas, noticed this strange numb patch on his arm years ago. Just a flat, discolored spot. Couldn't feel touch or temperature there. Looked a bit dry, no hair growing on it.
That hypoaesthetic, hairless skin plaque is very characteristic of tuberculoid leprosy. The sensory loss is key.
He finally showed it to his doctor years later.
Yeah.
They did a biopsy. And turns out it was tuberculoid leprosy, caused by Mycobacterium leprae.
The tuberculoid part refers to the type of immune response.
Right. The doctor explained that his body's immune system, specifically the cell-mediated part, driven by Th1 cells, had mounted a really strong fight against the bacteria.
Exactly. In tuberculoid leprosy, you have a vigorous Th1 response. These T cells produce interferon gamma, which activates macrophages very effectively to contain and kill the bacteria. This strong response keeps the bacterial load very low, hence the limited number of skin lesions and the relatively mild localized symptoms. The nerve damage happens, causing the numbness, but the infection is well contained.
So his immune system basically did a good job keeping it in check. The doctor said it was a testament to his strong immune response, but he still needed treatment.
Yes, treatment is still crucial to prevent further nerve damage and clear the infection completely. Usually multi-drug therapy with dapsone and rifampin for about 6 months.
It's quite remarkable, though, how the type of immune response dictates the severity. There's another form, lepromatous leprosy, with a weak Th1 response and much more severe disease, right?
Precisely. It's a spectrum largely determined by the host's specific immune reaction to M. leprae.
Fascinating. Okay, now let's explore why there's no effective vaccine for N. gonorrhea.
Gonorrhea, a persistent public health challenge. The core reason we lack a good vaccine boils down to this mechanism: antigenic variation of pilus proteins.
Antigenic variation.
Yeah.
Okay. I was talking to a friend who works in public health, and she was venting about how frustrating it is still no vaccine for gonorrhea. She explained that the bacteria, Neisseria gonorrhea, is like a master of disguise. It's constantly changing its outer coat, especially these little hair-like things called pili. They're essential for the bacteria to attach to our cells, right? Then they change.
Exactly. The pilus is a major target for our immune system, but N. gonorrhea has genetic mechanisms that allow it to constantly switch the version of the pilus protein it displays on its surface. It's called antigenic variation. So the immune system might generate antibodies against pilus version A today.
But tomorrow, the bacteria is showing pilus version B.
Precisely. Or version C or D. There's a huge repertoire of possible variations. This rapid switching allows the bacteria to effectively evade any immunity built up from a previous infection or, hypothetically, a vaccine targeting a specific pilus type.
So it's not like measles where one shot protects you for life because the virus doesn't change its coat like that. With gonorrhea, your immune system just can't keep up with the disguises.
That's the core challenge. It explains why people can get reinfected multiple times and why vaccine development based on targeting these variable surface structures has been so difficult.
Which makes prevention through other means, like condoms, and effective treatment even more critical. And the treatment itself is getting trickier with resistance, needing dual therapy now.
Right, often ceftriaxone plus azithromycin due to widespread resistance patterns. It's a constant battle on multiple fronts.
A real moving target. Okay,
Yeah.
next, cystitis and pyelonephritis by E. coli.
The urinary tract infections, E. coli is the king here, and a key virulence factor enabling this is Fimbria ramillispah.
Fimbria,
Hey.
like little fingers. My sister used to get recurrent UTIs. Awful. She described this terrible burning pain when she peed, constantly feeling like she had to go, and this deep ache down low in her belly.
Classic symptoms of cystitis, bladder infection, dysuria, frequency, urgency, suprapubic pain.
Sometimes, though, it would get worse. If it traveled up into her kidneys, pyelonephritis, she'd get a high fever, chills, and this sharp pain in her back right in her flank area.
That's the key differentiator for pyelonephritis. Fever, flank pain, costovertebral angle tenderness. Urine tests might also show white blood cell casts, which you don't typically see in just cystitis.
And the culprit was always E. coli in her urine cultures. The doctors explained that this particular E. coli has these tiny, sticky projections called fimbriae, or pili.
Yes, these act as adhesins. They allow the bacteria to bind very tightly to the cells lining the urethra and bladder, the uroepithelium.
So they can hang on.
Exactly. They resist being flushed out by the flow of urine. This adherence is crucial for establishing infection in the bladder and then potentially ascending up the ureters to the kidneys.
So those sticky fingers let it climb up. Makes sense why it was so hard for her to get rid of completely without antibiotics like nitrofurantoin or TMP-SMX for cystitis, or stronger ones like fluoroquinolones for pyelo.
Correct. Those fimbriae are a major virulence factor that allows E. coli to be such a successful uropathogen.
She learned all the prevention tips, hydration, voiding after sex, cranberry supplements sometimes. But yeah, those fimbriae make E. coli a formidable foe in the urinary tract. Now, sticking with E. coli, let's explore pneumonia, neonatal meningitis by E. coli.
Right, so E. coli isn't just about UTIs. In newborns, it's a major cause of really serious infections like meningitis and pneumonia. A key virulence factor here is the K capsule.
K capsule. Okay, here's where it gets really worrying. My little cousin was born a few weeks early. Shortly after coming home, she got a fever, was really lethargic, wasn't feeding well at all.
Concerning signs in a neonate.
Her parents rushed her back to the hospital, and the doctors found she had a severe infection, first pneummonia, then meningitis, all caused by E. coli.
The devastating diagnosis. E. coli, along with group B strep and listeria, are top causes of neonatal sepsis and meningitis.
It was terrifying. The doctors explained that in tiny babies like her, whose immune systems are still immature, certain strains of E. coli have this special protective outer layer called the K capsule.
Yes, the K capsule is typically polysaccharide. It acts like a shield, or maybe an invisibility cloak. It helps the bacteria evade the baby's underdeveloped immune defenses, particularly phagocytosis by immune cells and killing by the complement system.
Like a slippery shield, allowing it to survive in the bloodstream and cross the blood-brain barrier to cause meningitis or infect the lungs, often acquired during birth, right?
Yes, often acquired from the mother's vaginal or rectal flora during delivery. This K capsule is a critical factor allowing it to cause such invasive disease in vulnerable newborns.
Diagnosis involved a lumbar puncture for CSF analysis, in her case, blood cultures. She ended up in the NICU for weeks on really strong IV antibiotics like ampicillin plus cefotaxime or gentamicin.
Standard empiric therapy for neonatal meningitis, covering the likely pathogens.
Thankfully, she pulled through. But it was a stark reminder of how vulnerable newborns are and how these bacterial structures like the K capsule can be so dangerous. Okay, shifting gears slightly, chlamydia resistance to velactam antibiotics.
Chlamydia, a very common STI. Its resistance to penicillins and related antibiotics, the velactams, stems from a unique feature of its biology: lack of classic peptidoglycan due to reduced muramic acid.
Right, okay, so my friend was diagnosed with chlamydia. And I asked her, why didn't the doctor just give her penicillin or amoxicillin, something common? She said her doctor explained that chlamydia is sort of a weird bacteria. It doesn't have the typical thick, rigid cell wall made of peptidoglycan that most bacteria do.
Exactly. Chlamydia are obligate intracellular bacteria, meaning they have to live inside our cells. They had a unique developmental cycle too, but crucially, their cell wall structure is different. While they have some components, they lack or have significantly reduced amounts of muramic acid, which is a key building block of the classic peptidoglycan layer found in most other bacteria.
And bolectam antibiotics, like penicillin, work by specifically targeting the enzymes that build that peptidoglycan wall.
Correct. They inhibit the cross-linking of peptidoglycan strands, weakening the wall and causing the bacteria to lyse.
So if chlamydia doesn't really have that same wall structure,
Then the lactams don't have their target, they're ineffective. It's like trying to knock down a house by targeting bricks when the house is built mostly of something else.
makes sense. So instead, they had to give her a different type of antibiotic.
Yes, treatment relies on antibiotics that target other essential processes like protein synthesis. Macrolides, such as azithromycin, or tetracyclines, like doxycycline, are the standard treatments.
It was a good reminder that not all bacteria are built the same, and you need the right tool for the job. Understanding that fundamental biology, like the lack of peptidoglycan, directly guides the treatment strategy for chlamydial infections, whether it's urethritis, cervicitis, PID, or even neonatal conjunctivitis or pneumonia. Okay, let's talk about viruses now. Influenza pandemics.
Ah, the big ones. Pandemics, like 1918 Spanish flu or 2009 H1N1. The mechanism behind these major global events is RNA segment reassortment, antigenic shift.
Antigenic shift. Okay, my old microbiology professor explained this really well. She said flu viruses are kind of unique because their genetic material, their RNA, isn't one long strand. It's broken into segments, like chapters in a book.
8 segments for influenza A, yeah.
So sometimes, if two different flu viruses, say, a human flu virus and a bird flu virus, infect the same cell, maybe in a pig, which can be infected by both.
The pig acts as a mixing vessel.
Right. Inside that cell, when new virus particles are being assembled, those RNA segments from the two different parent viruses can get mixed up and packaged together in new combinations. That's like shuffling two different decks of cards together.
That's reassortment. And if this process creates a virus with a completely new combination of surface proteins, particularly hemagglutinin H and neuraminidase N, that hasn't circulated in humans before.
Then nobody has immunity to it.
Exactly. That major change, creating a novel subtype, is called antigenic shift. Because there's little to no pre-existing immunity in the human population, the new virus can spread rapidly worldwide, causing a pandemic.
It's not just a small tweak, it's a whole new disguise. Like taking Lego pieces from a car set and a plane set and making something completely new and unrecognizable.
A good analogy. This potential for reassortment and antigenic shift is why scientists constantly monitor influenza viruses circulating in animals and humans, looking for novel strains that could trigger the next pandemic.
Truly a dramatic example of viral evolution with huge public health implications. Okay, so if that's pandemics, what about the regular flu season? Influenza epidemics.
Right, the yearly outbreaks we're more familiar with. These are driven by a different, more gradual process. Mutations in hemagglutinin, neuraminidase, antigenic drift.
Antigenic drift. Okay. So every year, it seems like someone I know gets the flu, even if they got their flu shot or had the flu the year before. My doctor explained it as antigenic drift. He said the flu virus is constantly making small errors or mutations when it copies its RNA.
Exactly. Influenza's RNA polymerase is error-prone. It doesn't have good proofreading mechanisms. So small point mutations accumulate over time in the gene's coding for those surface proteins, H and N.
He described it like the virus putting on a slightly different colored scarf or hat each year. Not a whole new outfit like an antigenic shift, just minor changes.
That's a good way to put it. These small changes, this antigenic drift, mean that the H and N proteins gradually change shape over time. So the antibodies your body made against last year's virus or from last year's vaccine might not recognize this year's slightly drifted virus quite as well.
They still offer some protection, maybe, but not perfect.
Right. Immunity wanes because the target has subtly changed. This allows the virus to partially evade previously acquired immunity and cause recurrent seasonal epidemics.
And that's why we need a new flu shot every year. Because the vaccine is updated to try and match the strains that are predicted to circulate, the ones that have drifted.
Precisely. The annual flu vaccine formulation is a direct consequence of antigenic drift. It's an ongoing race to keep up with the virus's evolution.
Fascinating. A constant, gradual arms race between the virus and our immune system.
Okay.
Next, CNS invasion by rabies. Truly terrifying.
Absolutely. Rabies is almost universally fatal when symptoms appear. Its insidious journey to the brain involves a very specific mechanism. Binds to AC receptors or retrograde transport.
Dinese receptors. Yeah. My friend's cousin, he lived on a farm, got bitten by a stray dog. Didn't think much of it at the time, just cleaned it up. But a few weeks later, he started feeling strange, a weird tingling pain at the bite site. Then he became agitated, anxious.
Prodromal symptoms, often nonspecific initially.
Then it got much worse. He developed this intense fear of water, hydrophobia, terrible spasms in his throat whenever he tried to drink or even saw water. Also afraid of drafts of air, aerophobia, confused, hyperactive.
Classic neurological symptoms of rabies encephalitis. The pharyngeal spasms causing hydrophobia are particularly characteristic.
The doctors suspected rabies immediately. They explained how the virus, after replicating at the bite site in the muscle, had literally latched onto nerve endings there. Specifically, it binds to nicotinic acetylcholine receptors, AChRs, at the neuromuscular junction.
Using that receptor as its doorway into the peripheral nerve axon.
Exactly. And then it hijacks the nerve cell's own internal transport system. It uses motor proteins, specifically dynin, to travel backwards, retrograde transport along the axon, up the nerve, towards the spinal cord, and eventually the brain.
It's essentially catching a ride on the nerve's internal railway system, moving towards the central command center.
Walking backward along the nerves is how they put it. Once it reaches the brain and causes that encephalitis, it's basically unstoppable, leads to coma and death.
Which is why post-exposure prophylaxis, PEP, the rabies vaccine plus rabies immunoglobulin given promptly after exposure before the virus reaches the CNS is so critical and life-saving.
Yeah. His case was tragic because they didn't seek PEP in time. A truly horrifying example of a virus hijacking our own cellular machinery to reach the brain. Diagnosis often relies on finding meagre bodies, those inclusions, in brain tissue post-mortem. Okay, next, we're taking a deep dive into HIV infection.
HIV, the virus that causes AIDS. The core mechanism, the way it gets into our key immune cells, is crucial. Virus binds CD4 along with CCR5 on macrophages early, or CXCR4 on T cells late.
Right, so I've been living with HIV for over 15 years. When I was first diagnosed, my doctor explained how the virus targets my immune system. He said the virus has this surface protein, GP120, that acts like a key. It binds first to the CD4 receptor, which is found mainly on helper T cells, the commanders of the immune system.
But binding to CD4 isn't enough. It needs a second key, a co-receptor, to actually get inside the cell.
Exactly. Initially, especially during transmission and early infection, the virus strains that dominate tend to use the CCR5 co-receptor. This is found on macrophages, dendritic cells, and some T cells. These are called R5 tropic viruses. But over time, in many people, the virus mutates. It evolves the ability to use a different co-receptor, CXCR4, which is found predominantly on T cells. These are X4 tropic viruses.
And this shift in co-receptor usage from CCR5 to CXCR4 is often a bad sign, right?
Yeah, my doctor said that shift often correlates with more rapid disease progression. The X4 viruses seem to be more aggressive in destroying CD4 T cells.
That makes sense as they target the main population of T helper cells more directly. This leads to a faster decline in the CD4 count.
Which is exactly when my CD4 count really started to drop faster. And I got worried about opportunistic infections, things like PCP pneumonia when the count drops below 200, or Maycam below 50.
Those AIDS-defining illnesses that occur when the immune system is severely weakened.
Thankfully, the antiretroviral therapy, the heart cocktails, have been amazing. They target different steps in the virus's life cycle: reverse transcription, integration, protease activity.
The goal being to suppress viral replication to undetectable levels, allow the CD4 count to recover, and prevent those opportunistic infections.
Exactly. It's kept me healthy for years. A real testament to understanding the virus's mechanisms, like its entry strategy, to develop effective treatments. Okay, next up, the fundamental immune structure known as a granuloma.
Right. Granulomas are fascinating examples of the immune system trying to wall off something persistent. The mechanism involves a specific sequence of cellular interactions and signaling. Macrophages present antigens to CD4U and secrete IL-12A CD4U differentiation into Th1, which secrete IFN macrophage activation.
Okay, that's a mouthful. Let's break it down. My older cousin has sarcoidosis, mainly affecting his lungs. He describes it as his immune system going a bit haywire, creating these tiny knots of immune cells, granulomas. His doctor explained it starts when macrophages, which are like the scavenger cells of the immune system, encounter some kind of persistent stimulus, maybe an infection like TB or in sarcoidosis, something the body perceives as foreign even if it's not an infection. These macrophages engulf the material and then present pieces of it to CD4 plus T helper cells. While doing this, they also release a key signal, interleukin-12, IL-12.
That IL-12 is critical. It acts as a signal to tell the CD4 plus T cells, okay, differentiate into Th1 cells. Th1 cells are specialized for cell-mediated immunity, for dealing with intracellular pathogens or persistent antigens.
So IL-12 pushes them towards the Th1 pathway, and then what do the Th1 cells do?
The newly differentiated Th1 cells then release their own powerful signaling molecule, interferon gamma, IFN. IFN is the main signal that superactivates the macrophages. It turns them into larger, angrier cells called epithelioid cells because they resemble epithelial cells under the microscope.
Oh.
These activated epithelioid macrophages then cluster tightly together, trying to wall off the offending stimulus. Sometimes they even fuse together to form multinucleated giant cells. This whole organized collection of activated macrophages, epithelioid cells, giant cells, surrounded by lymphocytes, is the granuloma.
That's this whole chain reaction. Macrophage signals T cell with IL-12, T cell becomes Th1, Th1 signals macrophage back with IFN gamma, macrophage gets activated and forms a cluster.
Exactly. It's a feedback loop designed to contain difficult-to-eliminate threats. You see caseating granulomas with central necrosis, typically in TB, and non-caseating ones in conditions like sarcoidosis or Crohn's disease.
Even though it's a defense mechanism, those clusters can still cause problems and organ damage just by taking up space and causing inflammation.
Precisely. Diagnosis requires biopsy, and management involves treating the underlying cause if possible, or sometimes suppressing the inflammation.
Okay, let's turn to a hallmark of cancer. Limitless replicative potential of cancer cells.
Yes, one of the fundamental capabilities cancer cells acquire. This is tightly linked to telomerase activation and loss of cellular senescence.
Right. My friend's mom had a very aggressive lung cancer. And despite chemo, it just kept coming back. Her oncologist explained that cancer cells are incredibly stubborn because they've basically figured out how to become immortal in a cellular sense. He said our normal cells have protective caps at the ends of their chromosomes called telomeres. Think of them like the plastic tips on shoelaces. Every time a normal cell divides, these telomeres get a little shorter. Eventually, they get so short it signals the cell to stop dividing, that's cellular senescence, or even to undergo programmed cell death, apoptosis. It's a natural limit on replication.
A built-in clock, essentially, preventing uncontrolled proliferation.
Exactly.
Yeah.
But cancer cells, they often find a way to switch back on an enzyme called telomerase.
Which most normal adult somatic cells don't express much of.
Right. And telomerase acts like a maintenance crew, constantly rebuilding those telomere ends after each cell division.
So the telomeres never shorten enough to trigger senescence or apoptosis. The cellular clock is effectively bypassed.
Their clock just never runs out. This, often combined with mutations that disable normal cell cycle checkpoints, like those involving P53 or RB, gives them that ability to divide endlessly, limitless replicative potential.
It's a critical step in malignancy, allowing for the continuous growth and accumulation of mutations that drive tumor progression and metastasis.
And it's why treatments can struggle. Even if you kill off most cells, the ones that survive can just keep dividing indefinitely. Really highlights the fundamental biological changes in cancer. Now, related to that, let's explore tissue invasion by cancer.
Another crucial hallmark of malignancy, what separates a benign tumor from a cancerous one. A key mechanism enabling this is decreased E-cadherin expression.
E-cadherin, okay. My aunt had advanced colon cancer, and the doctors were particularly concerned because it had started invading the surrounding tissues. Her surgeon explained it using a great analogy. He said normal epithelial cells, like those lining the colon, have these molecular Velcro fasteners on their surface called E-cadherin.
Cadherins are critical cell adhesion molecules. E-cadherin specifically helps epithelial cells stick tightly to each other, maintaining tissue structure and integrity, like cellular glue.
Right. But in many cancers, especially carcinomas, which rise from epithelial tissues, the cancer cells lose the expression of E-cadherin. It's like the Velcro gets ripped off or stops working.
This loss of adhesion weakens the connections between the cancer cells and their neighbors in the primary tumor.
So they become loose.
Exactly. They can detach more easily from the main tumor mass. This, combined with other changes like increased motility and secretion of enzymes that degrade the surrounding tissue matrix, allows them to break through the basement membrane and invade into adjacent tissues.
That's what happened with my aunt. The cells lost their E-adherent glue, broke free, and started pushing into the deeper layers of her bowel wall, even towards nearby organs.
That invasive potential is fundamental to malignancy and metastasis. It's a key factor in TNM staging and determines prognosis and treatment strategy.
It really made her cancer much more serious, no longer just a contained lump, but actively spreading. A fundamental shift in cell behavior driven, in large part, by losing that critical adhesion molecule. Okay, let's move to some congenital heart defects. First, persistent truncus arteriosus.
Right. This is a cyanotic congenital heart defect where the mechanism is failure of truncus arteriosus to divide into aorta and pulmonary artery due to absent effective aorticopulmonary septum.
Okay, failure to divide. My cousin's baby boy was born with a bluish tint to his skin, especially around his lips and nails. Cyanosis. The doctors knew right away something was up with his heart. An echocardiogram confirmed persistent truncus arteriosis.
So normally during fetal development, there's a single large vessel coming off the heart called the truncus arteriosus. A structure called the aorticopulmonary septum forms and spirals down, dividing this trunk into two separate arteries, the aorta leading to the body and the pulmonary artery leading to the lungs.
But in this baby's case, that septum didn't form properly or was absent.
Correct. So the division never happened. You're left with a single large artery, the persistent truncus, arising from the heart and overriding both ventricles. There's also almost always a large ventricular septal defect, VSD, underneath it.
So one big pipe instead of two, getting blood from both sides of the heart.
Exactly. This single vessel receives mixed blood, oxygenated blood from the left ventricle and deoxygenated blood from the right ventricle, mixing through the VSD. This mixed, relatively deoxygenated blood then goes out to both the body and the lungs.
Which explains the cyanosis, the blue color. Too much deoxygenated blood getting into the systemic circulation.
Precisely. These babies are cyanotic from birth. They often also show signs of heart failure quickly due to the increased blood flow to the lungs. Clinically, you might hear a single-second heart sound because there's only one semilunar valve.
He needed open heart surgery within a few months. They had to basically construct a separate pulmonary artery and close the VSD. A really complex repair.
Yes, surgical correction is essential, usually in early infancy, to prevent irreversible pulmonary hypertension. A critical reminder of the precise choreography needed for normal heart development.
Okay, another one involving that septum, detransposition of great arteries.
DTGA. Another cyanotic condition presenting in newborns. The mechanism here is slightly different. Failure of aorticopulmonary septum to spiral.
Failure to spiral. Okay. My best friend's baby was born looking really blue, and giving him oxygen didn't seem to help much, which is super scary. The diagnosis came quickly. Detransposition of the great arteries. The cardiologist explained it like the main pipes leaving the heart were plumbed incorrectly.
That's a good way to think about it. So the aorticopulmonary septum does form and divide the truncus, but it fails to undergo its normal spiral twist. As a result, the aorta ends up connected to the right ventricle, and the pulmonary artery connects to the left ventricle. The connections are swapped or transposed. D refers to the position of the aorta being anterior and to the right.
So you get two separate loops.
Exactly. Two parallel independent circulations. Deoxygenated blood comes back from the body to the right atrium, goes to the right ventricle, and then out the aorta back to the body. Oxygenated blood comes back from the lungs to the left atrium, goes to the left ventricle, and out the pulmonary artery back to the lungs.
Oh, wow. So the oxygenated blood never gets to the body, and the deoxygenated blood never gets to the lungs, unless there's some mixing.
Precisely. Without some connection between the two circuits, like an atrial septal defect, ASD, VSD, or patent ductus arteriosus, PDA, this condition is incompatible with life after birth. The severe cyanosis, often unresponsive to oxygen, is the hallmark sign. You might also hear a single, loud S2 heart sound.
They immediately started him on a medicine, prostaglandin E1.
To keep the ductus arteriosus open, allowing some mixing between the two circuits temporarily. Sometimes a balloon atrial septostomy is needed too.
Right, and then he had the big surgery, the arterial switch operation, the Jettine procedure, where they actually cut the arteries off and reattach them to the correct ventricles.
The definitive repair, usually done within the first few weeks of life. A complex surgery, but with good outcomes now. Another example where a subtle error in developmental spiraling has profound consequences. The egg-on-a-string chest X-ray finding is a classic clue too.
Wow. Okay, next. TET spells in Tetralogy of Fallot.
Tetralogy of Fallot, the most common cyanotic heart defect beyond infancy. TET spells are frightening, acute episodes. The mechanism is acute episodes of cyanosis and hypoxemia, AG during crying, feeding, exercise, due to sudden pulmonary blood flow, often 2 degrees to infundibular spasm, and G right to left shunting.
Right. Tetralogy involves 4 defects, but the spells are key. My friend's daughter had TOF. When she would cry really hard or get upset or even just exert herself feeding, she would suddenly turn a much deeper shade of blue, especially her lips and fingers. She'd get agitated, breathe fast, sometimes even go limp. These were her TET spells.
They can be terrifying for parents. Tetralogy includes pulmonary stenosis, narrowing of the outflow to the lungs, a VSD, an overriding aorta sitting over the VSD, and right ventricular hypertrophy. During a TET spell, something triggers a sudden increase in the degree of obstruction to blood flow going to the lungs. Often it's spasm of the muscle below the pulmonary valve, the infundibulum.
So the already narrow passage gets even tighter.
Exactly. This sudden increase in resistance to pulmonary blood flow makes it much harder for the right ventricle to pump blood to the lungs. At the same time, the VSD provides an escape route. So the deoxygenated blood from the right ventricle preferentially shunts across the VSD into the left ventricle and then out the aorta to the body.
A massive increase in the right to left shunt.
Yes, leading to a sudden, dramatic drop in the oxygen saturation of blood going to the body, causing profound cyanosis and hypoxemia.
It was scary. They taught her parents to immediately pull her knees up to her chest.
The knee-chest position. That increases systemic vascular resistance, which helps to decrease the right-to-left shunting and encourage more blood flow towards the lungs. Oxygen, morphine, sometimes beta blockers like propranolol are used too.
She eventually had the full surgical repair, patching the VSD and widening the pulmonary outflow tract. But those spells were a stark indicator of the underlying physiology. The classic boot-shaped heart on X-ray is another clue for TOF. Okay, let's discuss Eisenmenger syndrome.
Eisenmenger syndrome represents the end-stage consequence of certain untreated congenital heart defects. The mechanism is a progressive cascade. Uncorrected LUR shunt, AG, VSD, ASD, PDA, A, pulmonary blood flow, pulmonary hypertension, A, endothelial dysfunction, irreversible pulmonary vascular remodeling, A, pulmonary vascular resistance, ARA, shunt, E, cyanosis.
Wow, that's a sequence. Okay. My neighbor, an older gentleman, had a heart condition since childhood. A big hole between his heart chambers, probably a VSD or ASD, that was never surgically fixed back then. For years, he seemed okay, maybe a little breathless sometimes. But then in his 40s, things changed. He started getting really short of breath. His fingers and toes started looking bluish and kind of bulbous, clubbed, always tired.
That's the typical progression. Initially, with a large VSD or ASD, you have a left-to-right shunt. Oxygenated blood from the higher pressure left side shunts over to the right side and goes back to the lungs. This chronically increased blood flow and pressure in the pulmonary circulation damages the small arteries in the lungs over decades.
Causes pulmonary hypertension.
Yes. Initially reversible, but eventually the pulmonary vessels undergo irreversible remodeling, thickening, scarring. This dramatically increases the resistance to blood flow through the lungs, pulmonary vascular resistance.
So it gets harder and harder for the right heart to pump blood to the lungs.
Exactly. Eventually, the pressure in the right side of the heart and pulmonary artery becomes higher than the pressure in the left side of the heart and systemic circulation. At this point, the shunt reverses. Deoxygenated blood from the right side now flows across the defect into the left side and out to the body.
So the shunt flips from left to right to right to left.
Correct. That's Eisenmenger syndrome. The onset of cyanosis, clubbing, and symptoms like worsening dyspnea, chest pain, or even fainting, syncope, signal this shunt reversal. You might also see signs like a loud P2 heart sound.
The doctors told him it was irreversible. The damage to his lung vessels was permanent. And crucially, they said he couldn't have surgery to close the hole now.
Right, closing the defect at this stage would be catastrophic. The right ventricle wouldn't be able to handle pumping against that extremely high pulmonary resistance without the shunt as an escape route. It would lead to acute right heart failure.
So the treatment now is just supportive, managing the pulmonary hypertension.
Largely supportive, yes. Medications targeting pulmonary hypertension, like endothelin receptor antagonists or PDE5 inhibitors, can help symptoms. In severe cases, heart-lung transplantation might be considered.
A really stark warning about the importance of correcting significant congenital heart shunts early before these irreversible changes set in. Okay, next, atherosclerosis. The basis for so much cardiovascular disease.
Indeed. It's a chronic inflammatory process. The mechanism is a well-defined stepwise progression. Endothelial dysfunction injury, macrophage accumulation, foam cell formation, fatty streaks, smooth muscle cell migration, extracellular matrix deposition, fibrous plaque complex atheromas.
Right. My dad had a heart attack a few years back, all stemming from atherosclerosis. The doctors explained it starts subtly, often with some kind of injury or dysfunction of the endothelium, the inner lining of the artery, maybe from high blood pressure, smoking, diabetes, or high LDL cholesterol.
That initial injury makes the endothelium sticky and more permeable. LDL cholesterol particles infiltrate the artery wall and become oxidized.
Then the immune system gets involved.
Yes. Monocytes are recruited to the site, enter the artery wall, and differentiate into macrophages. These macrophages avidly engulf the oxidized LDL, becoming bloated with lipid droplets. These are the foam cells.
Or foam cells, okay.
Accumulations of foam cells form the earliest visible lesion, the fatty streak. It's still relatively flat at this stage.
But then it progresses.
Yes. Inflammatory signals trigger smooth muscle cells from the deeper layer of the artery wall, the media, to migrate into the inner layer, the intima, proliferate, and start producing extracellular matrix proteins like collagen. This forms a more complex, raised lesion called a fibrous plaque or atheroma. It has a lipid-rich core containing foam cells, cholesterol crystals, dead cells, covered by a fibrous cap made of smooth muscle cells and collagen.
And these plaques are the problem.
They are. They can grow large enough to narrow the artery lumen, restricting blood flow and causing symptoms like angina, chest pain, or claudication, like pain on exertion. Or, more catastrophically, the plaque can become unstable. The fibrous cap can thin and rupture, exposing the highly thrombogenic lipid core to the bloodstream.
Which triggers a blood clot.
Exactly. Leading to acute thrombosis, vessel occlusion, and events like myocardial infarction, heart attack, or ischemic stroke. That's likely what happened to your dad.
Yeah. Now, he's super careful with diet, exercise, takes statins, aspirin, all aimed at managing risk factors and stabilizing those plaques. It's a lifelong battle against this inflammatory process. Okay, next, thoracic aortic aneurysm.
TAA, an abnormal dilation of the aorta in the chest. A key underlying pathological process, especially in certain conditions, is cystic medial degeneration.
Cystic medial degeneration sounds unpleasant. My uncle had Marfan syndrome, a genetic connective tissue disorder. He needed close monitoring of his aorta. The doctors explained that because of Marfan syndrome, the middle layer of his aortic wall, the media, was weak. The elastic fibers and smooth muscle cells were breaking down. They called this process cystic medial degeneration.
Exactly. Marfan syndrome, due to fibrillin-1 defects and other connective tissue disorders like Ehlers-Danlos syndrome, collagen defects, impair the structural integrity of the aortic wall, particularly the media. This degeneration leads to loss of elastic fibers, accumulation of mucoid material, hence cystic, and overall weakening of the wall structure.
So the wall gets weaker and stretchier.
Precisely. A weakened wall is less able to withstand the constant pressure of blood flow, so it gradually dilates, bulges outward, forming an aneurysm.
They watched his aneurysm carefully with regular imaging, CT scans, I think, kept his blood pressure tightly controlled with beta blockers.
Standard management. Control hypertension. Surveillance imaging to monitor size.
But eventually it got too big. The risk of rupture was too high. He needed surgery to replace that dilated section of his aorta with a synthetic graft.
So surgical repair, or thoracic endovascular aortic repair, DVAR, is indicated for large, 5.5 centimeter typically, or smaller in Marfan, rapidly expanding, or symptomatic aneurysms to prevent rupture.
Because rupture is catastrophic, right?
Yeah.
Sudden severe chest or back pain, shock.
Often fatal, so preventing rupture is paramount. Cystic medial degeneration is a key factor predisposing to TAA, especially in those with underlying connective tissue disorders.
A powerful link between genetics and vascular structure. Okay, finally, let's wrap up with myocardial infarction.
The heart attack. While atherosclerosis provides the substrate, the acute event, the MI itself, is typically triggered by this mechanism. Rupture of coronary artery, atherosclerotic plaque, acute thromboses.
Right, plaque rupture. My grandfather, long history of high cholesterol, smoker for years. One day, boom, the worst chest pain he'd ever felt. Crushing, radiating down his left arm, cold sweat, nausea. Classic signs.
Classic presentation of an acute MI, that sudden onset of severe substernal chest pressure.
He got rushed to the hospital. They told us a fatty plaque and atheroma that had been building up in one of his coronary arteries for years had suddenly ruptured or eroded.
That rupture exposes the thrombogenic material within the plaque's core tissue factor, collagen, to the flowing blood. This immediately triggers the coagulation cascade and activates platelets. Platelets adhere to the ruptured site, aggregate, and form a dense blood clot, a thrombus.
And that clot blocked the artery.
Often, yes. The thrombus rapidly grows and can completely occlude the coronary artery, abruptly cutting off blood supply and oxygen to the portion of heart muscle supplied by that vessel. It's like a dam bursting upstream, but here the dam forms suddenly,
Leading to ischemia and, if prolonged, irreversible necrosis, death of the heart muscle. Myocardial infarction.
Correct. The goal of emergency treatment is reperfusion, restoring blood flow as quickly as possible, either with clot-busting drugs, fibrinolysis, or, ideally, mechanically opening the artery with angioplasty and stenting, primary PCI. Time is muscle.
They got him to the cath lab quickly, did an angioplasty, gave him aspirin, other antiplatelet meds. He was lucky. That quick action saved a lot of heart muscle.
Prompt reperfusion is absolutely critical, and long-term management involves addressing the underlying atherosclerosis and preventing future events with lifestyle changes and medications like statins, beta blockers, ACE inhibitors, and antiplatelets. Ventricular arrhythmias are a major cause of death early post-MI2.
A dramatic culmination of that chronic atherosclerotic process, triggered by that acute plaque rupture and thrombosis, really drives home the connection. And that brings us to the end of another fascinating deep dive.